It's my pleasure to introduce Dr. Angela Donà, a marine biologist and conservationist dedicated to protecting Hawaii's native reef ecosystems from the growing pressures of nutrient enrichment, warming, and invasive species. Her current research examines the physiology and ecology of dominant invasive macroalgae, not to try to say the last name, and how these species alter the balance and productivity of coral reef communities. Angela uses experimental and field-based approaches, ranging from nutrient uptake assays to photosynthetic performance analyses to uncover the mechanisms driving algal overgrowth and reef decline. Guided by a commitment to conservation, her work seeks to restore resilience in native reef systems through applied data-driven research. She earned her master's degree from the Marine and Atmospheric Sciences College at Stony Brook University and her Ph.D. in marine biology from the University of Hawaii at Donà. Please join me in welcoming Angela to get started. Is it okay if I turn the light on?
Yeah. You'll see the slides better anyway. Thank you so much. So as Kayla said, I am a researcher. I am a postdoctoral researcher in the Limo lab, where I study how algae use light and photosynthesis to thrive or survive in their reef habitats. Much of my recent work has focused on invasive species, why they thrive in disturbed ecosystems, and what we can do to control them. And today, I'll share both the ecological insights and the applied side of my research. Okay. Well, I'm going to start giving a little bit of context. And the context is that a lot of our reefs right now are definitely in a state of change. Many of our native marine habitats are groundwater dependent ecosystems, or GDEs. And land-based pollution is causing profound changes in our GDEs. We're losing reef taxa at all levels, so coral, predators, all the way down to algae. And there are very few sites left on Oahu where only natives dominate the coastal landscape. And that is what the photo is representing. This is Diamond Head, and this is a photo I took about a year ago, where pretty much everything you can see there is native. I'm sure I don't have to tell any of you, but Limu is a very important member of the reef community. A lot of times you'll see people who study other things say that algae are killing corals or killing the ecosystem, and it's not really that's happening. It's an imbalance in the system. Limu are the primary producers. They're the base of the food web upon which everything depends. And they're deeply connected to the people and have high cultural value. They're used in ceremonial practices, as you know. We have 661 native species in Hawaii. 450 of those are reds. 137 of them are greens. 71 are browns. And we have 3 seagrasses. 13.2% of these are endemic to Hawaii. But we actually know very little about these. We only have DNA records for just under half of them. And physiological data is badly missing. We need growth, photosynthetic rates, nutrient uptake rates on so many of these species. So I put together a list of papers that do have this type of information. That's this table that I made right here. 8 papers that work on 9 species. This may not be exhaustive, but it comes pretty close. This is what we know about photosynthesis, growth, and, say, nutrient uptakes of our natives. And in the context of changing reefs, this is really concerning. So in 1990, Glenn et al. Did some research at the natatorium and found that over 70% of the algae on that reef was native Sargassum polyphyllum. These Lumukala forests provided structure, food, and shelter to fish and invertebrates. They made a list of what they found there. There were 29 native species and one non-native. That was Acanthophora spicifera. This, at that time, was not yet abundant. But in 2002, Coles et al. Did a study at 15 sites in Waikiki, and they found that of all of the other reported species that had been found there, 70 of them were gone. That's 30% missing. We don't know if they're gone forever. They're just gone from these areas. They looked at Aquarium Reef and Kaimana Beach Reef. That's this one up at the top. And here, these two are the most degraded, where invasives are dominating the reef. Lumukala forests were greatly reduced. And if you think about the types of algae that used to be there and what's there now, Gracilaria salicornia and Acanthophora, these do not form forests. They form mats and they stick close to the surface, and they do not any longer provide protection for fish. So if you go into the water here, you're not likely going to see the type of abundance of fish that you used to. So the real drivers of ecological change, not so much that algae are killing coral, but it's the imbalances in these ecosystems. So the conditions in which natives once thrived have changed. And imbalances favor things that grow quickly. Invasive species tend to grow quickly, and they're also free from predation. It's important that we address the reasons for the imbalances, the abiotic factors, as well as the factors that determine invasive algae dominance in these degraded habitats. Now, this is a map of Oahu, clearly. And what you see is a heat map, in the color of the red that you see on the map is basically showing us where we have a lot of on-site sewage disposal systems. And you can see that the area here, which is where we were talking about in Waikiki, that has a fair amount of that red color. We have 88,000 cesspools in the main Hawaiian Islands, and 50,000 of them are on Big Island. Cesspools are holes in the ground, basically underneath your house, where human sewage goes. And these discharge approximately 53 million gallons of untreated sewage into groundwater daily. They contaminate aquifers with high nutrients and anything else we put into our bodies. And then they mix with submarine groundwater and deliver contaminants to coastal marine ecosystems. But SGD, or submarine groundwater discharge, this is a natural phenomenon. It originates from rainfall that seeps through the porous basalt and delivers cold freshwater and naturally elevated levels of nutrients. The blue arrows on this diagram from Donà et al., those show natural pathways of groundwater that actually make their way into the ocean. SGD sustains native species in healthy habitats, but it is also a pathway for pollution. And that's what these orange arrows are showing. So cesspools that are in, near homes and other places of habitation, those are going into the groundwater and eventually making their way into our coastal ecosystems. And this contaminated SGD appears to be a major factor in invasive species growth. The federal definition of an invasive species is a non-native alien species whose introduction causes or is likely to cause economic harm environmental harm, or harm to human health. There are several dominant marine invasive algal species in Hawaii, and I have worked with 3 of them. Acanthophora spicifera, Hypnea muciformis. It is now called Hypnea caribica, but I am going to continue with muciformis because I will get it wrong for sure. And this is Chondria tumulosa. Currently considered a nuisance species because we don't yet know where it's coming from for sure. I think that's going to change very soon. So now I'm going to talk about the research we've been doing in using macroalgae as bioindicators. So we were investigating changes to the coastal ecosystem given climate change scenarios. We used growth of 2 co-occurring species as key bioindicators. That was Ulva lactuca, or Limu palahalaha, Hypnea muciformis, hookweed. Both of these exhibit invasive behavior on Maui and Oahu, but Hypnea has not yet been found in Kona. And that's a major factor in this research. So we have groundwater dependent ecosystems along the Kona coast. These are very important. They are sites of vibrant algae and coral populations. This map is from Johnson et al. In 2008, and what they did was aerial surveys that showed the colder water temperatures coincided with SGD pour points. So the red is warmer water, so oceanic water, and also low amounts of nutrients, but anything from, say, purple going up, which is you'll see in Honokohau Harbor, you can see that the temperature is really low there, and then as you move out towards the coast, it gets warmer because that is where it's mixing with seawater. So temperatures also coincided with elevated nutrient inputs, right? So also this graph at the bottom. And so as it mixes with seawater, those temperatures increase and then the nutrients disperse. We did an experiment that we wanted to use predictor variables that were chosen to simulate these conditions, say from oceanic conditions into the coastline, where it was heavily influenced by SGD. This is our site. The black dots on this map here represent where SGD enters the coastal ecosystems, and the orange areas are sites where we did field work to ground truth. So this is also a high priority area for cesspool conversion. And this map from Sea Grant and SOAS shows you that the Kona region, where we did our study right here, is a very high priority. Lots of cesspools here that are feeding really close to the coast. So we wanted to know, with this background, we wanted to know if macroalgal populations would change with changes to these GDEs. And those were based on predictions of reduced rainfall due to climate change. These would essentially result in less groundwater to feed the ecosystem and less seawater and freshwater mixing. We added on top of that scenarios of increased planned urban development. And this would, of course, draw more of the fresh water due to pumping for consumption. And then we also added mitigation factors such as native forest conversion or protection and wastewater treatment upgrades or lack of. This was our project workflow. So in the diagram top left, those are the scenario inputs. So climate, development, wastewater, and forest invasion. Those fit into the groundwater model, which was looking at things like salinity, temperature, and nitrogen, which is also similar to the marine water quality model. These are not the parts of the project that I worked on. This was a large collaborative effort. And what I did was the manipulative experiments that provided the growth data. And the growth data went into regression analysis, and we also added the field work that we did. And then together, these went into the spatial predictive modeling. All of this resulted in a paper that Brittany Okuhata led that came out in 2023. So what I can talk to you about a lot is the methodology for the experiments. So I am going to show you a lot of details about the physiological experiments that I did for various invasive species and other things. And I'm going to lay it out right now so that each time you'll know that this was the same basic experiment with maybe different variables. So all of them include an outdoor period of time where they draw down nutrients and they acclimate to that new irradiance. These are conducted as 9-day experiments. The predictor variables we use are salinity, temperature, and nutrients. We measure wet weight and photosynthesis on days 1, 5, and 9. And then we put the data into mixed effects models. So that's separate from the regression analysis. So this is where we conduct the actual experiment. Then they come into the greenhouse and get placed into individual jars, and the jars are in these bins. All of the plants are trimmed to a similar size. They're aerated separately. They go into filtered seawater or salinity dilutions. We change the water and inoculate, rotate the jars, rotate the bins every 48 hours. And then for this experiment, we used 6 salinity, nitrate, and phosphate combinations at 3 daytime temperatures. We had 48 individuals. There was 24 of each species. And each 9-day experiment yielded an N of 2. So that is low replication. Therefore, this experiment had to be repeated 12 times. Ultimately, we have a data set that had an N of 16, which is not too bad. So this is Limupolahalaha. This is our native chlorophyte. This is a great indicator of SGD. If you're in Kona, you can see there's a lot of really vibrant populations of this plant. It grows prolifically where nutrients are elevated. And if the tide is high, you can see turtles grazing. And when the tide is low, you can actually see a lot of the cropped plants. Hypnea muciformis is our invasive species, or hookweed. It's not yet on Hawaii Island. This was introduced intentionally in the early 1970s. It's hooks attached to substrate and also to native species. And it is highly successful in reproducing even as tiny fragments. Now, this is a photograph from the 1990s. There were persistent blooms in Maui near a wastewater treatment plant. And these blooms caused a tremendous amount of loss of revenue, estimated at about 20 million a year in the 1990s. So this would just sort of wash up on the beach and rot, and nobody wanted to be around it. Okay. So now I'm going to start showing you some data. Perfect, it's on this side. I'm going to orient you to these plots. You're going to see a few of them. The x-axis is always going to have our treatments, and the y is always going to show you growth, 9-day growth in terms of percentage. Here, on the left side, you have the oceanic conditions where you have your highest value of salinity, and in SGD, you have your lowest value of salinity. This is 11 parts per thousand, and here's 35. Then oceanic conditions also have the lowest amount of natural nitrate. This is 0.5 micromoles. In here, we bumped it up to 80 micromoles. It's a lot. But that is a natural value. These were the values found by Johnson et al. In 2008. And so what you see is that ulva really likes to be in SGD. does really well, even though the salinity is quite low, it does really well with all of that nitrate. So linear increase, the mean is about 50% growth. Hypnea is a different story. So what we found here is that if you keep 35 parts per thousand, but you increase the nitrate, you get an increase in growth. If you keep, well, actually, we doubled the nitrate and we lowered the salinity, we got a decrease in growth. Then keeping that salinity the same and increasing the nitrate, an increase in growth. But from there, we basically saw decreases in growth. So it looks like salinity is a problem for this plant unless it gets a fair amount of nutrients. But this is the important one right here, that even though it has the same amount of nitrate as the one to its right, it does less well when you decrease that salinity. So this is a really important key for us to understand sort of what drives this plant. And yeah, comparing the SGD treatment for Hypnea, here you can see we only have about an 11% mean growth in Hypnea. So that data then goes into the predictive models, but first they go into these boosted regression trees to determine the power of each of those predictor variables. So you can see nitrate was the strongest predictor for ulva. And then nitrate, salinity, and temperature were roughly equal as predictors for Hypnea. Then we combined the land sea models, that data for Kona, and then the output of these spatial distribution models for each of these scenarios showed that habitability for Hypnea generally increases. That's the dark red color that you see on the map, not the pink, so you'll see more dark red. So the first panel on the left is climate change relative to the baseline, which is now. The second panel is urban development relative to climate change. And then the third is native forest conversion plus urban development. Native forest conversion means no protection. So we're seeing an increase in habitability for Hypnea, whereas the opposite is what we're seeing for ulva. So ulva's habitability actually decreases for each of these scenarios. That's not good. So we know that ulva thrives in SGD. Now, well, you could see it, but now we have data. And that Hypnea may be limited to higher salinities. These models basically found that climate change reduced rainfall is going to favor Hypnea growth along the Kona coast. So we have to hope that it never gets there. We wanted to take this a little bit further. I did some more experiments using wastewater levels of nitrate, because if you think about some of those, some of the values that you see here, I didn't show a map necessarily of Kona, but you know, we do have a lot of wastewater going into the ecosystems. So we decided to run some more experiments. And the question really was, is there a true salinity threshold for Hypnea? And would large nutrient subsidies override this effect? And what we found is that Hypnea does quite well in high nitrate. Now we lowered the salinity to 18 at the most. And we used nitrate values from 80, which was the highest value for SGD. So we started from that point and went up to almost 3,000 micromoles. And this is because this is from the predictors actually at Honopohau Harbor. So salinity, we did 18 and 22 parts per thousand, and we had a little bit of overlap with our SGD work. We found that we got over 100% mean growth in ulva, and Hypnea growth increased as well. So what you're seeing here now, the same type of graph, only I've added to the right the wastewater data, and the SGD data, that's the red box, that's the data that I just showed you for Hypnea. And you can see the overlaps are the green means that is 18 parts per thousand, and you can see that also is in the wastewater. So we did 18 parts per thousand. As you add nitrate, you get higher growth. Then if you compare, say, this one here, the 11 parts per thousand in the SGD, and compare that, so now you've got two that have 80 micromoles of nitrate. Those, you can see that if you increase salinity, then you get higher growth. So basically we found that salinity it has an effect, but that nitrate is definitely a strong driver here. We got over 50% mean growth for Hypnea in the wastewater treatments. But as you can also see, even if we went up to 3,000, there's not that much difference. Okay, so now I'm going to move on to the work that we did in the monument, invasive seaweeds in the Papahanaumokuakea Marine National Monument. I do believe it has become a sanctuary. Yeah, are we saying sanctuary now?
National Marine Sanctuary.
Yeah. Yay, yeah. Yeah. Okay, it's going to still say monument because I've just been very careful. I went onto the website and it still says monument, so I was like, okay. So sadly, Acanthophora spicifera was recently found up at Koihilani. And the managers up there had contacted our lab to see if we could find a way to stop it from spreading. And now we have this methodology to run tests, to try to understand it, hopefully find a weakness that we can exploit. We also got some tissue samples from them that showed they have some sort of elevated nitrogen source. We don't know exactly where it's coming from. This is the site Rusty Bucket where they collected the Acanthophora. It's just out on the reef there, and it's at the end of an old runway on Sand Island. So Acanthophora spicifera, its common name is prickly seaweed. It's abundant on Oahu and other main Hawaiian islands. It was introduced unintentionally in the 1950s. And it's commonly found in areas with freshwater input and nutrient subsidies. This traps a lot of sediment, if you've gone to the beaches and see it, it traps a lot of sediment. And it grows long and weedy. It has a superpower. It makes a lot of branchlets. We ran the same experiments on this species. We used our best guesses at what their local ecological conditions would be so that we could run experiments that represented the place. We used two temperatures, 24 and 28. That's basically their coldest and their warmest temperatures throughout the year. We ran two salinity and nutrient combinations. We did an oceanic, just like in the SGE treatments. So high salinity, low nitrate. And then we did a moderate decrease in salinity to 28 with a pretty decent increase in nitrate to 14 micromoles. And what we found here is that the basal tissue was usually just kind of died off, but a lot of little apices grew. So the weight in itself was not a good parameter to look at this to see what was going on. And I'll show you here. Both of these treatments here, if you look at 0% growth right here, you can see they're hovering around 0% growth. That's because as they were losing tissue, they were gaining tissue. And that's for both treatments. But this graph is showing us the number of secondary apices, right? And so the oceanic treatment had a mean of around 40, but the bumped up treatment where you're getting a little bit lowered salinity, but higher nitrate, you're getting, I think, around 60, 65 as our mean. Just explain secondary. Yes, secondary apices. I will show you.
Sorry.
Well, actually, oh yeah, you want me to? Sorry to all of those of you on Zoom. It would be any of the growth that would come off of here, right? So this will be the main axis. Actually, I'm going to show you. Well, we do think that the growth of those apices, and the fact that they're going to just become new little plantlets, that's a really good way to understand the success of this plant. Okay, this is, here we got some secondary apices, right? So this is one of the plants that was involved in the experiment, and all of these are secondary apices. And they're super furry. And because they're super furry, that doesn't mean that it's just one little plantlet that's going to come off of here. This main axis is dying. You can see here there's a bend. That's not natural. That's because it's actually going to be lost. We're going to lose all of that main axis tissue, and we're going to have 6 new plants. In fact, you can see here, I hope, that, below the line, that's the tissue we're going to lose, and then we're going to have at least 1 new plant. This is what Acanthophyra can do. We wanted to know, okay, so what happens next? We get all of these new plantlets. How long will it take for them to attach to new substrate? So I set up a new experiment. This is different. This one here, I provided a few types of substrate. I did sand, coral skeleton, and bio-rock of opportunity. These were, you can't collect bio-rock, but these were used in another experiment under permit. And what we did is we used them also for this experiment. I provided filtered seawater, but I did not aerate. We wanted these to have a little bit of, still water so that they would have time to settle. Checked them at different intervals, at 24-hour intervals, starting at 48 hours. And I would basically just sort of probe them with forceps just to see if they would move or if they would stick. I confirmed attachment to the bio-rock after 72 hours. And after 14 days, 35 of the 48 individuals were attached. 13 were not, and 11 of those 13 were to that coral skeleton. So we think that the fast growth of these numerous secondary apices, and then the break off and become these new little plants, and then the rapid attachment, we think this has a lot to do with this plant's success. But it doesn't really stop there. One of the main parts of the mandate for this work was to find a way to kill it. And that's tough in the marine environment, but we needed to try. So we have lots of this plant, and I ran it through several different types of trials. All of them would be environmentally unproblematic, things that we could do in the water. First thing would be desiccation. Second thing, I soaked them in Dawn dish detergent for 30 minutes. I know. I know it sounds funny. Then desiccated, then rehydrated. Then I soaked it in baking soda. Desiccated, rehydrated. And the way that we evaluated whether these were, whether they worked or not, was by looking at fluorescence. And this is a proxy, it's what we measure when we're measuring photosynthesis. We were looking for a complete lack of fluorescence. 0 fluorescence would mean it was dead, and any fluorescence on that plant would mean it is not dead. This was the only one that worked or had any promise. So baking soda looked like a good way to continue. Around the same time I was running these death trials, we started talking to the Chondria tumulosa working group. So this is the, a group of federal, state, also PMDP, the Papahanaumokuakea Marine Debris Program, and other scientists. So the main concern now with chondria is it's spread to the main Hawaiian Islands. PMDP goes up to the monument and does missions where they pull up a lot of marine debris, nets, ghost nets, anything that doesn't belong up there. If they find a net that is visibly entangled with chondria, they are not allowed to bring it back. This then stays there in the environment and continues to be a danger to all of the flora and fauna. But the ones that they are allowed to retrieve, they keep these in large shipping containers on the ship. And as of right now, the best management practice tells them that they have to soak these in 10% bleach solution. This is kind of dangerous. They don't like to do it. It sloshes around a lot. It's not very safe on a rolling ship, and it's not something you really want to be dumping into the ocean. Not at least at those quantities. So what they were hoping to find is a way to kind of bypass this and kill chondria without having to do the bleach. And they were hoping it would be desiccation because that would be the cheapest way for them to move forward. So we were going to give this a try here on island, and we cannot use chondria here because, well, it's not here and we can't bring it here, but we have a ton of Acanthophora, and Acanthophora is a very similar, it's very closely related. So plentiful, we use this as a proxy. We tested the efficacy of desiccation using net and debris filled bins. Then if this was going to work, then we could then sort of scale this up to shipboard containers. We took 10 algae packets and embedded them in the debris. There were 6 bins of each type. One set was aerated, one was not. And then we also put humidity and temperature loggers in them. We ran this for 5, 14, up to 60 days at the Hawaii DAR facilities at Sand Island. And just if you're wondering, 5 days was the amount of time that it takes PMDP to travel back from Kwahilani down to Oahu. So they were hoping that this would be sufficient time for it to desiccate and be dead. It did not, desiccation did not in these bins kill Acanthophora. We measured the fluorescence, and again, if we found any fluorescence, it wasn't dead, and therefore, even one plant within a bin of 10 would mean that that treatment is not successful. And this is really important that we set this high bar for success so that the unthinkable doesn't happen. None of the plants, in fact, were dead in the aerated or non-aerated bins. And also, it was hard for us to achieve full desiccation in these bins. They weren't really drying out that well. Imagine how hard that would be in a shipping container, with the just tremendous amount of debris that they're bringing up. Okay, so now I'm going to move to talk more about chondria tumulosa. This was first reported in 2016 at Manavai, and it rapidly spread throughout the northern atolls of the monument. So it went to Kwahilani and then eventually also to Holaniku. This forms large mats that detach from the substrate and float. They likely raft to new habitats. This is what we think is the likely way that it got from, say, Manavai over to Kwahilani and then further on. This is Taylor Williams, one of Heather Spalding's students, together with a big mat of chondria tumulosa. It's huge, right? And so this is a problem. So in 2021, it was discovered at Kwahilani. This is a map from Chelsea Council at NOAA. They did surveys of the lagoon around the atoll, within the atoll, and the far, let me see, right over here is where we do our collections. They found over 40% of that area in 22 and 23 was chondria. It's possibly more than that at this point. There are a lot of labs that are working on this species. They're working on eDNA. They're trying to figure out where it came from and also some phylogenetics. But you cannot work on this species if you're looking to do ecological or physiological experiments unless you go there. And we were very, very lucky to be able to travel to Kwahilani and do some work there. So we brought our traveling laboratory there. This is where we filtered seawater. Let me see. Okay, left. Top left is where we filtered seawater. Center top is the cube where we were shading and running the experiments. The bins will look similar to the ones that we did at AFRC. And then this looks just like we were running in the lab here. And then Celia Smith and I got to do this. That's us also collecting irradiance and water samples. This is a photograph I took of chondria in April of 2024. And I think the first thing you can see about this is that it has two colors. Never mind the green, those are, that's another species. But the golden part is the canopy, rich in carotenoids, gets a lot of sun. And then the purple part, that is the understory. That's the part that actually attaches to the reef. There are some challenges to working at Kwaihelani. We had 1 month of time to do this. We had no time to acclimate, and we had very little experience with this. So we had to make some really, quick decisions about what variables we would use in order to have the most replication and learn the most in a shorter period of time. And we thought that separating the canopy and understory was going to be really important to do. We chose 2 salinities. These are the same that we did for Acanthophora. We did 4 moderate nitrate and phosphate levels. We went up to 8 micromoles, which is pretty moderate considering some of the other values I've told you about. And we were unable to really control the temperature, so this was ambient temperature. We ended up with an N of 12, not too bad. So again, same type of data I'm showing you. Again, treatments on the bottom, we went from oceanic 0.5 micromoles all the way to 8 micromoles. Canopy, understory, these are separated out. And also the salinities, you can see those are binned for each of these nitrate values. And I think that the interesting thing here, or not interesting, is that there are no trends. Even though we gave 8 micromoles of nitrate, we would have expected to see a lot of growth in this plant, and we did not. Not in the canopy and not in the understory. What's going on there? But we did see changes in pigmentation. So when you put them into an experimental condition from out in the ocean, the light changes. And what we saw was that within days, the pigments changed. So the canopy became a little bit darker, more understory-like. It also, it tends to be very fragile, very brittle. So when you touch it, it breaks. These broken fragments did not tend to survive. So that's very different from, say, Hypnea, and also really different from Acanthophora. The understory instead, it decreased in pigmentation from those phycobilins and went more towards the carotenoids. But this also started to change morphologically, and it grew more branchlets, more canopy-like. It became this, so they sort of swapped. So trying to make sense of this data, we think the plasticity in the pigmentation and morphology is going to help those rafting mats as they're traveling from, say, one place to the other, because they're going to roll and they have to survive a, who knows how long journey. So this is important for spreading from place to place. Those inputs of 8 micromoles, that was a surprise. It really should have induced growth, but it didn't. So either the plant is highly efficient at nutrient uptake, or the ecosystem is nutrient replete. And in the monument, these are not the typical degraded ecosystems that we have here. So this is a bit of a mystery. We really don't know what's going on yet there. We think, though, that there's efficient nutrient uptake. And so this must be a major factor in the rapid spread. Okay, winding down, we also did some depth trials on these, trying to figure out, now that we were there, we could actually work with chondria. So we tried bicarbonate because it had been very promising with Acanthophora. Nope, this did not work. And I'm going to skip through all of the other ones and just get to the point here. None of it worked. We did try at the very end of our trip in 2024, tap water and seawater heated to 50 degrees, and this showed a lot of promise. But we did have to leave, and we didn't have a lot of time to sort of like test that any further. So in 2025, we did some more hot water trials, and we found that seawater and freshwater with replication, this works if you heat it to 60 degrees. So we can start to plan what would be the next steps for PMDP when they go out and they collect these nets. And the hope is that we can figure something out that they can go next year and start collecting these nets that are out there. I think this is really exciting. So for chondria, we do hope to return so that we can continue to better understand this species. I think it is critical to remove biomass. There is a lot of it, and even though it will be messy and it is very pricey, I hope we can find a way to do this. Concern about the fragmentation has been a deterrent to action, and I think that's valid. But unlike Acanthophora and Hypnea, those small fragments do not appear to be a mode of dispersal. So worry is valid, but if we don't do anything, we are sure to fail. For the native algal population, we need a lot more data. I showed you, we just don't have a lot of information on our native algal species. And if I look at this, and I hope we can think of this in a very positive light, that if we start to change the problems in the ecosystem, we'll be ready with a lot of data on our natives, and hopefully we'll be able to see them return. Cesspool conversion is mandated by 2050. I hope that we find a way to get that done. It's really expensive for homeowners to do it. There's a lot of people who cannot do it. And so we have to find ways to help. And if we want to see that done, because it's a major cost and it's really hard. So we know that these ecosystem imbalances have allowed at least Hypnea muciformis and Acanthophora to thrive and dominate in our ecosystems here in the main Hawaiian Islands, but it is unclear what's going on and how this is happening in the sanctuary with chondria. And our data is showing that the current ecosystem imbalances are going to continue to favor an increase in these invasive species. I hope that if we can make these changes, that the natives will return. So I'm just going to finish with this quote here by a former Hawaii DAR administrator: "You cannot manage what you do not understand." And I think that's important as we're all trying to grapple with what do we do about these problems. I might also add, you cannot protect or restore what you don't understand. So there's a lot of work on algae, and this is such an important, I think, topic. And I hope this has been interesting for you, and that it has hopefully inspired some of you to do some work in algae. Who knows? Big thank you, board, as I finish. And yeah, let's see if you guys have some questions.